to discuss is nitric oxide. So this truly is an unconventional neurotransmitter in that it's a gas. It's not a solute that can be dissolved in an aqueous medium. It's not a peptide, but rather it's a gas. And nitric oxide is produced from the metabolism of the amino acid arginine, as is illustrated here in this pathway. So arginine is processed by an enzyme called nitric oxide synthase, and nitric oxide in a gaseous form is produced, as is citrulline, and that nitric oxide then is free to diffuse across the plasma membrane, or it can interact with target structures, maybe within the cell where the gas was synthesized. Here in this particular illustration, we're showing the activation of a guanylyl cyclase enzyme, which results in the production of cyclic GMP, one of the important second messenger systems we have within the brain. Now, one of the interesting activities of nitric oxide involves its ability to diffuse freely from, let's say, a postsynaptic cell to a presynaptic cell. So there's ongoing interest in the idea that nitric oxide might be some kind of a retrograde messenger that allows from signals to travel back from a postsynaptic cell to a presynaptic site. Of some interest is the activities of nitric oxide as a vasodilator. And so this is a function that is performed in a variety of tissues, and that activity is mediated by a guanylyl cyclase pathway. And perhaps it's of interest to some of you that Viagra and other related drugs that are used to affect erectile dysfunction function by affecting nitric oxide mediated vasodilation. Similar pathways, however, are found in a variety of structures within the brain and elsewhere in the nervous system. Hence, these drugs can often have side effects, often involve sensory processing. And we'll see why when we talk about the mechanisms of phototransduction, and we'll discover an important role for the guanylyl cyclase pathway within the photoreceptors of our retinas. Okay, well, let's look now at a side-by-side comparison of the processing of small molecule transmitters and peptide transmitters. So I would encourage you to have a look at the table that I've prepared for you in in page 3 of your handout, which talks about the synthesis, the packaging and release, and the removal of neurotransmitter at synapses that use small molecules and peptide transmitters. So with respect to the synthesis, small molecules are synthesized typically in the presynaptic terminal, where there are enzymes that are present that can create the active neurotransmitter right there in the presynaptic terminal. For peptide neurotransmitters, the synthetic pathway is primarily in the cell body, where the peptide is processed like all other peptides through the endoplasmic reticulum, the Golgi apparatus, where various kinds of post-translational modifications may be performed. Meanwhile, this peptide can be packaged into a vesicle of some sort and then transported down the axon. And along the way, further modifications or perhaps cleavages of that peptide can occur as the initial product of the transcript is then processed and prepared for its active function in the presynaptic terminal. A couple of other differences to note. Small molecule transmitters typically are packaged into vesicles that then are concentrated right along the active zones at the presynaptic membrane, whereas peptide transmitters are often found in vesicles that are diffusing around the presynaptic terminal further away from that active zone. So consequently, the vesicles that contain the neuropeptides, which typically are much larger than those that contain the small molecules, are further removed from those active sites and therefore require a larger influx of calcium in order to trigger calcium-mediated fusion events. So with a relatively modest depolarization of the presynaptic terminal, we're likely to only see the calcium-mediated fusion of small vesicles that release the small molecule transmitters. It's only with a very large depolarization that the presynaptic terminal is likely to release much peptide neurotransmitter, because in order for that to happen, sites well away from our typical high-efficiency active site are engaged, and those larger vesicles then fuse and release peptide neurotransmitters, but only when that presynaptic terminal is flooded with calcium. So this might be one physiological way of holding in reserve the peptide neurotransmitters only when there is intense depolarization of the presynaptic terminal. And lastly, considering the removal of the neurotransmitter once it's been released, the small molecule transmitters have a variety of systems. The transmitter itself might be degraded, and precursor substances might be then taken back up into the presynaptic terminal, or they may run through a glial cell where they may be in some storage form before being returned from a glial compartment to the presynaptic terminal. Of course, the transmitter can simply diffuse away as well. For a peptide neurotransmitter, these substances likewise are free to diffuse away, but there are also proteases that are found here in the synaptic cleft that can cleave these peptide neurotransmitters and degrade them right at their region of release, in and around the synaptic cleft. So let's also take a few minutes to consider a typical glutamatergic synapse in the brain. So as I've already mentioned, glutamate's one of the most important small molecule neurotransmitters that we have. It's widely considered to be the most important small molecule for mediating fast excitatory events in the central nervous system. So in a typical presynaptic terminal, we have an important enzyme called glutaminase. And this enzyme converts glutamine into glutamate. And glutamate is then concentrated in synaptic vesicles, where it's taken into that vesicle via a transporter called vesicular glutamate uptake transporter, so VGLUT for short. So this synaptic vesicle then, of course, can fuse with the presynaptic terminal when calcium floods that active zone. The glutamate is free then to diffuse into the synaptic cleft, where it can interact with postsynaptic glutamate receptors. Now, some of this glutamate, of course, diffuses away from the synaptic cleft, where there are excitatory amino acid transporters in the glial cells that surround the synaptic compartment. And of course, the important glial cell that we're talking about here would be the astrocyte. And so the excitatory amino acid transporter takes up the glutamate, where it's converted back to glutamine. So glutamine is, in essence, a storage form of glutamate that can sit in reserve within astrocytes before being recycled as needed back to the presynaptic terminal. So there are other kinds of co-transporters that allow glutamine to cycle back to this presynaptic terminal, where it can once again be hydrolyzed by glutaminase into glutamate, ready for another round of synaptic release. Okay, lastly, let's just make a couple of points here in conclusion. So we've talked about our two classes of neurotransmitters, our small molecules, our peptide transmitters. We've also talked about what we've called some unconventional neurotransmitters. And these neurotransmitters interact with two classes of receptors, our ionotropic receptors, which mediate rapid synaptic events, since the receptor for the ligand is part of an ion channel complex, and there are metabotropic receptors that require the activation of second messenger systems. So they mediate more slowly evolving and potentially longer lasting synaptic effects. Our small molecule transmitters can interact with both kinds of receptors. Our peptide neurotransmitters, as far as we know, only interact with the metabotropic family of receptors. Now, I think from this simple schematic, it should be apparent to you that if we want to understand the postsynaptic activity of a neurotransmitter, we need to know more than simply the identity of that transmitter. It's not enough to know if it's a small molecule or if it's a peptide. So knowing the chemical structure itself is not sufficient to understand the physiology of the effect mediated by that transmitter. One needs to understand something about the receptors that interact with that neurotransmitter in question. So in order to account for the physiological activity of a chemical neurotransmitter, you need to know the receptor. So in our next tutorial, we're going to talk more about the structure and function of both types of receptors, the inotropic receptors and the metabotropic receptors. And I think you'll be impressed, as I am, with the diversity of effects that can be activated, even by the very same chemical signal. So next time we'll talk about neurotransmitter receptors. I'll see you then.